Great. Thanks for the organization this morning and a pleasure to be here. So we're going to talk about silicon effects improving stress tolerance of floriculture crops. We've done some case studies with salt and drought and heat, and then a little bit of work with disease that we'll talk about as well. And I wanted to note we've been talking about how some species are silicon accumulators and some aren't. So I wanted to show a little bit of our preliminary work, which back in the day was to see which floriculture crops kind of took up silicon, and then if there were kind of effects on their quality. So we started with this experiment conducted by my postdoc at the time, Roland Leatherwood, where we had our tap water, which we tested. It had, or the tap water had very little silicon, zero point 0.8 parts per million. The substrate itself, which was a peat-based potting mix, had 2 parts per million silicon. And we did weekly drenches of just over 110 parts per million silicon. And we grew these 21 different floriculture species. We sent them to a laboratory for analysis, and of these 21 varieties that we looked at, there was a range of accumulation. So the highest accumulators had about 5,000 parts per million silicon, which is about 0.5% by dry weight, similar to the amount of calcium or phosphorus in these crops. And some of the higher accumulators were Terrenia, Argyranthemum, Verbena, and Calibrachoa, and New Guinea impatiens. And then the shaded bars represent the silicon concentration without the addition. So all of these species naturally were able to glean some silicon from the soil and from the water that was our source. And then these white bars represent the concentration with these once weekly silicon drenches. And so an interesting thing that we found was about half of the species we looked at did not differentially take up silicon. So maybe they didn't have the underlying machinery to absorb additional silicon. And then about half of the species, the ones that tended to be higher silicon accumulators, seemed like they did have the genetic machinery to take up more silicon. And in some cases, it was 50% or 100% higher in the tissue. I wanted to point out Petunia because we'll show you a few pictures of Petunia later on with silicon responses. Petunia was a low silicon accumulator, but it did differentially take up silicon. So when we drenched the plants with silicon, they took up about twice as much. And so that to us stood out as kind of an interesting model plant that had low amounts in the tissue, but did seemingly absorb additional silicon when we added it. Um, and this was just another way to look at the data. So for about half of the species that I mentioned, we did see a significant increase in the percent of leaf silicon content. So that varied from about 10% to about 140% for these accumulators. And then these were the species that showed enhanced leaf silicon. So Argyranthemum, Calibrachoa, both garden impatiens as well as New Guinea impatiens, lobelia, garden geraniums, petunias, Sutura, Terrenia, and Verbena. We measured the plants kind of at the time of harvest. So we looked at different horticultural traits or quality characteristics such as height or plant width or diameter of the stem base, the fresh weight and the dry weight, the percent water content, the flower size, and then the leaf size. And where we found statistically significant differences, the positive means that silicon enhanced that trait. So we had, say, greater flower diameter. And the negative means that we saw slight reductions in that trait. So slightly shorter plant or a slightly smaller flower diameter. What I will say is these were not huge effects. So if you just visually looked at the silicon treated plants versus not, you probably would not have noticed visually these differences. And then one other interesting thing to find out is The plants that were silicon accumulators were not necessarily the ones that showed positive or negative results. So sometimes there were crops like scavola or fuchsia, which were not silicon accumulators, but they did apparently have some horticultural trait effect. And the way that we summarized this research was that weekly silicon supplementation increased leaf silicon concentration for about half of the species studied. There were some differences sometimes for cultivars within a species and the growth effects of silicon. Differential silicon accumulation was not required to necessarily see that growth effect. So even those plants that had low amounts of silicon and didn't necessarily seem to accumulate more in their tissue showed a response. And then we did not see any overall dramatic effects on plant growth. And as we kind of since learned more about silicon, we realized that silicon really affects plant stress responses, whether that be insects or disease or abiotic stresses. So at that time, then we decided to start stressing plants, trying to kill plants, which we love to do because we don't have to sell them. So we had a study where we had 28 bedding plant species, again, planted into a peat perlite potting mix. And then we had treatments where we gave them added sodium chloride. So this was a salt stress experiment. And 40 millimolar sodium chloride, that's about 1,000 parts per million sodium and about 1,200 parts per million chloride. Which is more than 10 times the kind of recommended amount that we would have in our water source. And then they received plus and minus about 50 parts per million silicon as part of their constant liquid feed. And New Guinean patience was one of the 8 species that showed a significant response where the control plants that were not stressed with salt, did not necessarily show a silicon effect. But then when the plants were stressed with that 1,000 parts per million of sodium and chloride, if they did not have silicon, those plants were dead by the end of the 5-week study. And if they did have silicon, while the plants were not maybe marketable or saleable, they were alive and growing. And this was again a pretty severe salt stress, so I would not expect to see this much salt in the irrigation water, but it was a way to stress the plants. So under maybe more of a moderate salt stress, we would see kind of better recovery effects. So for 8 species, and I'll show you that list in a second, the salt-stressed plants that had silicon were significantly larger than their counterparts that had salt stress, but not silicon. And if we kind of look at that for New Guinean patients, when the plants didn't have salt stress, we did not see an effect whether they did not have silicon or had silicon, but when they had that high amount of salt stress, the plant biomass or dry weight was significantly greater when they had silicon. We did not see any cases where silicon reduced the growth of unstressed plants. And then we saw 4 additional species that showed some benefit, but it was not statistically significant. And that list of plants that showed a benefit from silicon in terms of salt was Bacopa, fibrous begonia, calibrachoa, kufia, dahlia, impatiens, pansy, and snapdragon. So then another line of research that we did, and Mohammed showed or alluded to this example, is we produced poinsettias and then with and without silicon as part of their constant liquid feed program. And again, that would be about 50 parts per million silicon every day. That was from potassium silicate. And then we did not see at kind of harvest time or what we'd consider shipping time, there was not major differences between the plants. So silicon didn't have a negative or positive effect just on the way the plant looked. But then we brought the plants into a simulated post-harvest environment. So we brought them into a room that was at room temperature with fluorescent lights on for 12 hours a day. So maybe simulating what would happen in a big box store where the poinsettias get shipped out there, but then not watered. And we noticed that the plants that did not have silicon during greenhouse production, they wilted earlier. So this was 12 days post-harvest. So at 12 days, our silicon-treated plants are fully hydrated. Our plants that did not have silicon were wilted. And then they recovered better from wilt as well. So we watered wilted plants and then looked at their recovery, and the plants treated with silicon recovered better than their counterparts without silicon. So this could be kind of a tool in the toolkit in terms of improving post-harvest performance. Another way that we've looked at stressing plants and seeing whether silicon had an effect was with petunias. And we became interested in petunias, like I said, because they were not a huge accumulator of silicon, but they did um, uptake silicon or have higher leaf tissue concentration. So we grew poinsettias in a greenhouse with and without weekly silicon drenches. So at this point, we drench them once a week with 100 parts per million silicon from potassium silicate. And then those plants were moved to growth chambers that we had at either 95 degrees Fahrenheit or 102 degrees Fahrenheit. We kept the plants fully watered at this point, so the wilting that we're seeing is not a drought stress per se. And so what we saw was that our silicon treated plants were not as affected by that heat. They stayed fully erect and they did not shut down. That was at 95 degrees Fahrenheit. At 102 degrees Fahrenheit, which is a little bit more severe of a stress, they're still looking better than their counterparts, although they would not necessarily look marketable. Um, and then this is 3 days at 102 degrees Fahrenheit, where again our silicon treated plants kind of recovered during that heat stress and didn't look as bad as after just a day. One of the phenomenon that we noticed, so this was a follow-up experiment where we treated petunias with this would relate to about 25 parts per million silicon or 100 parts per million silicon. And then before or after the heat stress, we took leaf tissue samples and we sent them to a lab for analysis. And so even when there was 0 parts per million of added silicon, there was some silicon in the leaf tissue. But then if we added silicon as part of the constant liquid feed program, we saw we saw higher amounts of silicon in the leaf tissue. But the crazy thing to me is after the heat stress, the leaves had much higher concentration of silicon in their tissue. And so we call that phenomenon stress-induced silicon uptake, where the plant is seemingly responding to that stress by telling the roots to absorb more silicon and transport it to its leaves. And then I had to include a picture of the Finnish flag here because if anyone knows Finnish culture, there's this term called sisu, which means apparently it doesn't directly translate to English, but it means stubbornness and resilience. And so silicon improved the resilience of petunias and gave those plants sisu, as the Finns would say. So what about insects and disease? When there's quite a bit of literature, scientific literature on silicon improving insect and disease resistance. And there's kind of two modes of action that are talked about, or two kind of large buckets of modes of action. And one of those is direct effects. And so for those plants that are higher silicon accumulators, verbena is one of them, sunflowers, cucumbers, grasses. That silica actually gets deposited in the hairs of the trichomes, as well as in the surrounding cuticle or the kind of layer of the plant that's the skin of the plant. And so then the thought is that these kind of glassy silica deposits can interrupt pathogens the spore might be trying to germinate and penetrate into the cell. Or in the case of insect feeding, it can make insect feeding more difficult. For example, wearing down the mandibles or the jaws of an insect, for example. And then the other kind of bucket of modes of action as indirect effects. And so these might be plants that are not high silicon accumulators, but silicon seems to affect gene signaling. Sometimes the stress hormones like jasmonic acid are increased. And then that kind of turns on plant defense responses. So the plant kind of prepares for the attack, and silicon helps mediate a quicker defense response. This is just an example from the literature that's kind of a classic picture of the direct effects of silicon. And so what we're looking at are the mandibles or the jaws of armyworm that had been feeding on corn or maize leaves that had been treated with silicon. In this particular study that was from 2003, they found that the mandibles were intact and not as worn down if the corn plants did not have supplemental silicon, and then if the corn had supplemental silicon added to the fertilizer program, that means there were more of these glassy silicon deposits in the leaves, and so the mandibles were worn down as the armyworm was chewing through the leaves. And then in this particular work, they also found that it reduced the fecundity or the number of offspring that each armyworm was able to have. So basically, it wasn't able to get as much nutrition from the leaf as it was feeding on it. Jennifer had a really nice example with powdery mildew and zinnias, where there was nice effects on powdery mildew suppression. We had a project some years ago that was a collaboration with Dr. Marjorie Daughtry at the Long Island Horticulture Research and Extension Center, where we wanted to see if silicon improved tolerance to Pythium root rot. In this experiment, we had New Guinean patients and snapdragons. We were looking at Pythium irregulare, which is a common species of Pythium that affects both of those crops. And then we had 6 different treatments in this study. So essentially minus or plus Pythium inoculation. So this is where Marjorie grew plugs of Pythium in agar, and then we inoculated the root zone with those. And then we had kind of 3 different silicon situations. So we had a set of plants that did not receive silicon, but were plus and minus Pythium inoculation. Then we had a set of plants that got that drench or liquid silicon, that was potassium silicate, that was part of their daily constant liquid feed. And then we had a set that had substrate incorporated silicon, and that was about 200 grams of silicon per cubic yard with a potassium silicate source. And unfortunately, we did not see maybe huge effects. So this row of snapdragons on the back is the ones that were not inoculated with Pythium. And then the ones in the front were all inoculated with Pythium. The one on the left did not have any added silicon. The one in the middle had the liquid silicon every day, and then the one on the right had substrate incorporated silicon. And one of the gleanings that we made at this time is it could be that the amount of Pythium that we inoculated the plants with or infected it with was a higher amount than what the plants would kind of naturally get from natural Pythium pressure. And so maybe that overwhelmed the ability of silicon to help improve some of those disease tolerance effects. About halfway through this experiment, I remember scoring the amount of wilting that the snapdragons had, and the silicon treated plants had a little bit less wilting than their counterparts that didn't have silicon. So there was some evidence that there was an effect of silicon with that study. And then for New Guinean patients, similarly, if the plants were inoculated with Pythium, we didn't necessarily see a silicon benefit in that particular study. And you can see the roots of the plants that had been inoculated on the right side versus plants that were not inoculated with Pythium on the left side. So maybe that was just a little bit of a cautionary tale. I think there could still be more room to explore Pythium resistance and silicon, but silicon's not a miracle cure for every plant and every particular disease organism, and depending on the pressure of that disease organism. Just a few practical notes before we go to some questions. So if you're using a liquid silicon source, for example, the potassium silicate that I've used, um, this particular one, potassium silicate, cannot be mixed in the same stock tank as our standard water-soluble fertilizers that we use for greenhouse crops. So the things like the calcium nitrate and potassium sulfate and so on, those can precipitate out. And what this is showing is kind of the jar test that Sean was actually talking about. I guess I did this in a beaker because I'm a scientist, but essentially we took the silicon potassium silicate at 100 or 200 parts per million silicon and combine that in the dilute form with the water-soluble fertilizer. And we did not see any precipitation at kind of the dilute amount that the plants are getting. But once we had those in the concentrated amount, such as going through a 1 to 100 injector, that's where we got that sludge or precipitate that Sean was referring to. So those weren't compatible, and then that would not be able to be taken up by the injector and distributed to the plants. So if we're using this potassium silicate source, we would not be able to mix it in the same stock tank as our standard fertilizer. Ideally, we would have a second fertilizer injector running in series. So the first injector could pick up our standard water-soluble feed, dilute that down, and then the second injector can pick up the silicon source and dilute that down. And at that dilute amount that the plants are getting, they can be mixed together. The other alternative is to do a once weekly drench when you're not fertilizing. So you could switch the injector stock tank line over to a different stock tank for that once per week. Um, and my suggested trial rates, which I highly recommend doing your own in-house trials with a small number of plants, um, but would be to use potassium silicate at 50 parts per million silicon if you're using it as a constant liquid feed, or if you don't have that second injector, then use it at 100 parts per million silicon as a weekly drench. In some of our studies where we've looked at the cost of adding silicon to the fertilizer program, adding that liquid silicon for about a 5 week period of time added about 1 cent per pot to the cost. And if it was a longer crop like poinsettias, it would add about 2 cents per pot to the crop. The substrate incorporated silicon that we used for that one trial was a significantly cheaper way to add silicon. So that came out to about 0.2 cents per pot for adding that substrate incorporated silicon. And it is a slightly alkaline material. So in our studies, we see that if we're adding potassium silicate to the fertilizer program, it raises the pH by about 0.3 units over the course of a 5, 6 or so week production period. So that might start to, sometimes for plants that are sensitive to iron deficiency, like petunias or calibrachoas, we might have to monitor those a little bit more closely and be ready to do acid drenches if pH is getting too high. So my overall thoughts on using silicon based on the greenhouse research that I've done is that the addition of silicon to greenhouse plants growing in soilless substrates appears to be beneficial for certain plants under certain conditions. And so especially if a stress is present, we have often seen improved stress responses with silicon. Also, depending on the tap water source or the fertilizer source or the substrate, it might already have sufficient silicon in it. So to see a further silicon benefit, that would imply that those sources don't have enough silicon already in them naturally. Peat-based potting mixes have very little silicon, so some of the potting mixes that are more like rice hull based or could be a better substrate source. And then silicon doesn't appear to be a miracle cure for every stress, for every crop. We have not really seen any negative effects of silicon in our research. I remember some cut flower studies that I was reading in the literature, and at very high rates of of silicon, much higher than what we were using. They occasionally saw for like sunflowers or zinnias, which are very high accumulators. They saw slightly smaller flower size, and they related that to maybe the mechanical effect of those silicon deposits made it hard for cells to expand. Okay, then before we go into questions, we have the poll, which will be the last poll that you'll have to take today
All right,
to prove
so
that you stuck
thanks,
with
Neil.
us.
I will launch the final poll.
Beautiful.
Okay, so folks should see that on the screen. And again, if you need those pesticide applicator credits, this is the final poll that you'll have to participate in. These polls aren't anonymous, so I can check who has participated. Question is, how has your knowledge of supplemental silicon changed following today's presentations? Select one. So increase greatly, moderately, some, or no change. Okay, I'll leave it up for a few more seconds. I think everyone has participated. I'll end the poll and share the results. So everyone can see the results.
Yay!
All right,
Not
I
so
know.
bad. Yeah,
But yeah, for the most part,
some knowledge
50% said increase
increases. Yeah.
greatly and then 38% moderately. So hooray to all the presenters. All right, I'll stop and I'll let you continue.
Great, cool. So I just wanted to acknowledge funding sources for the research that we did, which were the New York Farm Viability Institute, the American Floral Endowment, and the Gleckner Foundation. And this is what AI thinks I do for a living. So I'm a furry orange Maine Coon cat that hangs out in the greenhouse and does research. All right, with that, I'll look at the Q&A and happy to take any questions. So. Um, let's see. One that came up from Melanie is that poinsettias sometimes require PGRs to control plant height and stretch. If silicon increased height in sunflowers, has the same effects been seen in poinsettias? So in our poinsettia trials, which were fairly rigorous, well, we had two cultivars of poinsettias. We repeated it over 2 years, but we did not see effects on height from silicon. And I would say it's, in my experience, pretty unusual that we see height effects. Let me go back to our slide that had these 21 floriculture species. So we had three pluses here. So apparently for portulaca, lobelia, hot water blue, and impatiens pure beauty white, we saw slight increases in height. And then for bractanthia, we saw a slight decrease in height. But otherwise, for the rest of those 21 species, we did not see effects on height. So overall, I don't believe for poinsettias you would need to adjust your PGR program. And then what element is the potassium silicate precipitating with? I should know that, but I'm going to guess, which I probably shouldn't do, but I'm thinking it's probably the phosphates or the sulfates that are common in our complete water soluble fertilizers. So we typically have like magnesium sulfate or potassium sulfate, and then we have like monopotassium phosphate, for example. So I think it is those things. And when in doubt, you can always do the jar test that Sean talked about, is mix the two fertilizers together in the concentration that you're thinking about using them, and see if a precipitate forms. I also like to let that jar set overnight to see if a precipitation formed after 12 hours or so.
Neil, I have a question for you. Is silicon highly mobile within the plant?
It is. There's a whole molecular biologists love this. So in some of the monocots that are known high accumulators, like grasses and rice and corn, for example, they've actually found the transporters responsible for taking up silicon monosilicic acid by the roots. And then for offloading that, so it goes up the xylem, and then it's offloaded by the xylem. And then it's not phloem mobile. So once it's been deposited in a leaf, it can't be reabsorbed by the plant. So for example, nitrogen is phloem mobile. So if a plant is deficient in nitrogen, the plant can scavenge nitrogen from the old leaves and ship it up in the phloem to the new leaves. But the plant can't do that with silicon. So it's kind of like copper or kind of like copper or boron or calcium in that regard.